Welcome! Today, we're going to tackle one of the coolest puzzles in all of immunology, how our body, with a limited genetic playbook, can possibly prepare for a literally infinite number of enemies. So let's get right into it. But you know what? First, a little immunology humor to get us started. Why don't anteaters get sick? Because they're full of antibodies. Okay, bad jokes aside, let's get down to business. So here's the big paradox we're diving into. Think about it. Our entire human genome has what? Maybe 20,000 to 25,000 genes in total. But the number of different viruses, bacteria, and other pathogens out there is just staggering, practically endless. So how in the world does our immune system get ready to fight invaders it's never even met before? Well, the solution obviously can't be one gene for one bad guy. Our bodies just don't have that kind of storage space. It would be genetically impossible. Instead, our immune system came up with this absolutely genius system that uses a limited set of reusable parts to build an almost infinite variety of detectors. The best way to think about this is like having a giant box of Legos. You don't need a unique brick for every single thing you want to build. You just need a versatile set of building blocks. For the immune system, these are little gene segments called V for variable, D for diversity, and J for joint. And here you can see what that box of Legos looks like inside our actual DNA before any of the assembly begins. This is what's called the germline configuration. All those potential V, D, and J pieces are just sitting there, all lined up in a row, waiting to be picked. Okay, so we have the pieces. But how does a cell actually pick and choose which Legos to use to build its one-of-a-kind receptor? This is where the magic really happens. Let's look at the incredible process that literally shuffles the genetic deck for every single immune cell. The process is called somatic recombination. You'll also hear it called BDJ recombination. And it's the absolute engine of all this diversity. It happens inside our developing B cells and T cells, and it's how each and every one of them gets a totally unique antigen receptor, different from all its brothers and sisters. It's basically a three-step process, a kind of molecular cut-and-paste job. First, the long strand of DNA literally bends into a loop to bring the chosen pieces right next to each other. Then, a special enzyme complex called VDJ recombinase, which is made of the famous REG1 and REG2 proteins, acts like a pair of molecular scissors and snips the DNA. Finally, the cell's own everyday DNA repair kit comes in and pastes the ends together, making a brand new unique gene. And this visual right here, it's perfect. It shows you exactly how that looping and cutting works. See how these gene segments that were far apart on the straight DNA strand are brought together? The whole loop of DNA in the middle with all the pieces we didn't use just gets chopped out and thrown away, leaving us with a new, shorter, and totally functional gene. Okay, listen up, because this is super important, especially if you have an exam coming up. The massive diversity we're talking about comes from two main places. First, you have combinatorial diversity. That's just the simple math of mixing and matching different V, D, and J segments. But the second source, junctional diversity, is where things get really wild. Here's the crazy part. The way these pieces are stitched together is intentionally a little messy. Enzymes can chew away a few nucleotides, and another enzyme, TDT, can come in and just add totally random ones. This creates a ton of variation right at the joint, in a spot called the CDR3 region. And why does that matter? Because the CDR3 is like the very fingertips of the receptor. It's the main part that actually touches the antigen. That little bit of planned sloppiness is the secret to getting a truly insane number of unique receptors. So, okay, we have this amazing system for creating millions of different receptors. But wait a second, how do we know they even work? And maybe even more importantly, how do we stop them from getting confused and attacking us? Well, that requires a super strict quality control process. Let's start with B cells. This whole maturation process happens in the bone marrow, and it's like an assembly line with checkpoints. First, the cell builds its heavy chain. And this next step is a huge checkpoint, a real make-or-break moment. The new heavy chain has to prove it can pair up with a temporary partner called a surrogate light chain. If it can, it forms something called the pre-B cell receptor. Passing this test sends a powerful signal to the cell: survive and make more copies of yourself. It's a way to make sure the hardest part is working before the cell wastes energy making the final light chain. Now, T cells go through a really similar drill, but they do it in a different organ, the thymus. They also have a pre-receptor checkpoint.
Their newly made beta chain has to successfully pair with a stand-in alpha chain to form the pre-TCR. Only the cells that pass this test get to move on to the double positive stage, where they start expressing both CD4 and CD8 and get ready for their final exam.
Okay, so this is where things get really interesting, especially when we compare their final exams. Both cell types go through something called negative selection. This is simple. If a cell reacts too strongly to our own body's tissues, our self-antigens, it's dangerous and it gets deleted. But T cells have an extra test called positive selection. They have to be able to recognize our own body's special ID cards, the MHC molecules. If a T cell can't read those ID cards, it's completely useless for surveillance, right? So it's eliminated. It fails the test. B cells, on the other hand, don't have this requirement.
So after all that genetic shuffling, all that testing, and all that quality control, what's the final result? What does this whole process actually produce?
All right, let's wrap this up with the key takeaways. If you remember nothing else, remember this. 1. Diversity comes from somatic recombination, cutting and pasting V, D, and J segments. 2. You get a massive repertoire from both combinatorial and junctional diversity. 3. The cells go through checkpoints, like the pre-BCR and pre-TCR, to make sure they're not duds. And 4, there are 2 final tests. Positive selection for T cells only makes sure they're useful, and negative selection for both makes sure they're safe.
This whole system is it's beautiful. It's the bedrock of our entire adaptive immune system. The ability to create an army that's ready for any possible threat while making sure that same army doesn't turn against us is just one of the true marvels of biology. But what happens when that safety check, when negative selection fails, when a self-reactive cell slips through the cracks? Well, that's the origin story for autoimmune diseases, but that's a story for another day.